All right, I'd like to welcome everybody to the Pennsylvania Biotechnology Center. This is our 9th annual Entrepreneur Spotlight. This is one of my favorite events. It's where we get to showcase 3 of our PABC companies. We've got great panelists. We have a couple speakers we're going to get to first. But I want to take special mention. This is the home of the Hepatitis B Foundation and the Blumberg Institute. I think Randy Heyer, the president of the Blumberg Institute, is here in the crowd. Sherry could not make it today. I'd also like to welcome the founder and president, and one of the reasons why we're here, fresh off the year at Yale, writing a book on the Biotech Center, we're Dr. Timothy Block. Although he couldn't make it, I'd like to send a welcome from Congressman Fitzpatrick's office. Brendan McCluster is here. Thank you for the Congressman's support. Thank you so much. Yeah, our first speaker is one of our biggest champions. Quite simply, and I mean that we wouldn't be here today without the support, the grants that help build this beautiful new building, and of course, the state funding that allows the PABC, our 2 other organizations, to provide valuable services, research, and help us support the PABC community. It's my honor and my privilege to introduce our state senator, Senator Sanaciro.
Thank you. Good afternoon, everyone. This is a rare occasion. This might mark the first time that I've been at one of these events on time. So I'm very happy about that, excited. Of course, we had the governor here just a few months ago, as you know, and it was a great opportunity for him to see firsthand all the exciting things that are happening here at the Biotech Center at Bloomberg and at the Hep B Foundation. And I want to echo Lou's words. I want to thank Randy Shary, of course, who is not here today, Lou, and of course, Tim. As I said earlier, what a legacy this institution is for him. But I'm so grateful and honored that this is here in Bucks County and here in the 10th Senate District, because this really is a model for the entire state, which is something we wanted to convey to Governor Shapiro when he was here back in the springtime. And what you're doing here today, make no mistake, is a critical piece of that. Because by providing the capital, the resources, the investment in these companies that are here in Pennsylvania, to grow here in Pennsylvania, you're helping all of us in our, I think, shared mission of making Pennsylvania a leader in the biotech sector. And that's certainly something that I think was clear when he was here, is a goal that the governor shares. And I'm hopeful as we move forward past this year's budget process and into next year's future budgets, that we are going to make some substantial progress on pretty significant investments at the state level in making it happen. We have our, the other states that are competing with us, we know well what they're doing. And this is a foot race that we cannot dare fall behind in. They are making substantial investments. We need to equal, if not exceed that. And I'm confident that we will be in that position in just a couple of years. And again, what you're doing here and the example you provide for the rest of the state is critically important in that shared goal and that shared project. So I want to thank you, Lou. I want to thank everyone who's here today and participating once again this year for being here, and I'm excited to hear the outcome. So thank you.
Second speaker, I don't see Commissioner Harvey in the room, so if he does show up, I'd love to have him say a few words. He's been a big supporter. So we'll go right to our host in a second. I do want to make one note about what the senator's been able to provide along with Representative Bradford. We were awarded last fall the first ever pilot $5 million academic innovation zone grant. That allows us to provide at this site here, and Jeff Devano from Brandywine's in the back room there, down at B Labs, $2.5 million of funding each site to help out with free lab space, employees, equipment, IP. We're getting companies from across the country interested in coming to our 2 incubators, thanks to the efforts of Senator Sanaciro and Representative Bradford. So thank you so much for that opportunity. So I'm going to introduce our MC. I've done this so many times, I'm trying to come up with a new way to do it. Not too many, but I will say it's very important. The more time I spend with Grant, just his role at Sentinel Therapeutics. So he is the SVP of Commercial Strategy and Operations, but in discussions, he literally runs a worldwide international operation, and he covers everything from A to Z. And over the last several years, whenever we need somebody who's knowledgeable, who has great experience and knowledge of our industry, and the most important thing is he works for free. We call Grant. So, ladies and gentlemen, your MC for the rest of the evening, Grant Castor.
I'm gonna compliment Lou's shoes. He's got some like, hey dude, executive. Those are pretty good. So, good to see a packed house here. Thrilled to be here. We have an excellent agenda this afternoon. So, like I do every year, I want to highlight some key trends that are happening in our marketplace. So, in the last 12-ish months since we last met, according to one source, actually, let me back up. According to one source, year to date, we've only had 7 IPOs. Comparing that to 2022, in which there was 47 IPOs that only raised about $4 billion. Compare that to 2021, when there was 152 offerings when the market was hot, which raised a little bit more than $25 billion. So could biotech CEOs be missing the boat this year by not IPOing? So if we just look year to date, the NASDAQ is up, any guesses? 36.1% year to date. S&P 500, come on, any financial planners in the house? 17.8%, good guess. And the Dow Jones only up about 4.3%. So, year to date, life sciences announced M&A transactions, both from the private and public sector that have disclosed value, are representing about $84 billion in aggregate considerations. So it's likely, small, mid-cap M&A activity continues through 2023, as many big pharmas need to fill some pipeline gaps in the coming few years. So year to date, the FDA has approved, any guesses how many drugs? 27. Which puts them on pace to potentially beat last year's number of 37. However, it's far behind what they did 2 years ago. Often, in this space, we get solely focused on drug therapies, on what drug therapies get approved or transact. We often miss some other spaces, which I'm starting to become more aware about, and I think we're going to hear some of these spaces this afternoon. So first, med device, NuVasive to Globus for approximately $3 billion announced. Also the diagnostic space really heating up, saw a few $1 billion announced transactions this year. Services is a huge and growing sector that include many entrepreneurial opportunities. So think about clinical trial support, drug development services, such as the significant onslaught of AI, and I use quotations because a lot of it's just machine learning, that claim to be expediting the development timeline and ability to reduce the inefficiency of approximately 90% of drugs or new chemical entities that fail before they even reach the clinic. Regulatory and real-world evidence, CDMOs, and many others. In my PE circles I travel in, we have seen a huge uptake in PE groups making bets in the service sectors, and they're willing to deploy capital there because they actually have true balance sheet fundamentals, unlike early stage microcap biotechs and call it pre-publicly traded biotechs. So last year when we were here, many of us thought we were entering into a recession. Now, many analysts seem to be conflicted if we have achieved a soft landing or if we're about to experience a delayed recession. So putting the public markets aside, I am personally bullish on the ability to raise capital in the coming year and the continued appetite for M&A as a significant amount of dry powder is sitting on the sidelines right now, and especially in the private markets. So with that, I'd like to go ahead and introduce our distinguished panel. So all the way to the left, we have Greg Anthus of Tunnel Consulting. In the center, Jennifer Griffin of Mission Bio. And all the way to the right, Lorenzo Pellegrini from the Hatch Bio Funds. So each entrepreneur will have strictly, I say strictly, 10 minutes to present their company. And once they are done presenting, we'll pause for a brief Q&A. If they go over on time, we'll certainly limit the Q&A. So Greg, pass the mic to you to introduce our first organization and entrepreneur.
Thank you very much. So I'm pleased to introduce our first speaker, Dr. Kumar Shalabhai, founder and CEO of Boost Bioscience. Dr. Kumar has had over 30 years of experience in big pharma and biotech startup, and currently a senior advisor and partner here at the center. This is his third startup, the first two were successful and launching others on NASDAQ. First, we do a patent on a therapeutic application, alternative routes of administration for immunotherapies, oral, nasal, and inhalation, which many folks thought was not possible. And I was fascinated in our discussion last week about how he persisted and found a way, a new way to do this. So it was really fascinating science. I was involved by it. It's been validated by clinical studies and other products, and he has over 40 patents issued, so very prolific in terms of discovery and patenting. Passionate about this new class of therapeutics and is looking at the potential to replace current first-line therapies or therapies that are really not effective in the area, and can make this available to patients that don't have alternatives where we're all here. So welcome, Dr. Kumar Shalabhai, to the stage.
Thank you, Greg, for a nice introduction and also for kind words. I would also thank the organizers for giving me this opportunity and grant. I am targeting the missing space, pulmonary fibrosis. So, and you will see that in my presentation that the approach that we are going forward with is completely disruptive or innovative technology. With that, I chose this picture as background because I was in Greece a couple of weeks ago. And I love this picture because it's rising sun. And what I'll show you today, that new biosciences could very well be a rising star. And with that, let me move on to the first slide. Okay, this is a very important slide, which I also know nobody reads, so I'll move forward. This is our strategy, very simple. Simple in the sense that we will take compounds or drug candidates and deliver in such a way that it can have local action. By doing so, you're reducing the dose, maximizing clinical benefits, and reducing tox. And another good thing about our strategy is that we are using existing drug candidates. And those two candidates that we will talk about were discovered by. And we took those compounds with oral administration all the way up to phase 2. Therefore, those two compounds are completely clearless. I am using those two drug candidates for oral and inhalation delivery. Of course, I don't have time to go for the third compound, nasal administration, in the next 10 minutes, but I can tell you the nasal administration is also equally impressive. I'm also using AI, artificial intelligence, for selection of patients, as well as for biomarkers, and possibly down the road as surrogate biomarkers. These are the investment highlights, which of course are important only for the individuals here, that I have had experience of bringing compound from the lab to market. A drug that is already approved for 2 indications, flecainide, was invented in this center. And we took that compound all the way up to FDA approval. And by the way, I must tell you, by doing so, we employed 120 people. This company started with only 2 people, 120 people. In 2016, after the drug was approved, I also wanted to pursue another innovative or disruptive technology, that is to give antibodies through alternative routes of administration, that is oral nasal, and inhalation. Nobody had used this alternative routes of administration before. So my patents were first in the field. What I'm pleased to tell you is that oral administration and nasal administration have now been clinically successful. And we also use inhalation and validated the technology with monkey study. So that is something very exciting for us. New Biosciences is targeting autoimmune diseases. Particularly, I believe, following this COVID-19 pandemic, there could be tsunami of pulmonary fibrosis. And our approach, as you'll find out when I go in details of the technology, is unique and it has great potential to be successful. We filed patent applications. I don't know if Nick is here, but he was very helpful in filing this patent application. And this patent application covers our technology. As I said, the program is de-risked, but for some reason portions are deleted or not shown here. But both atipramab and dolkenatide have been through phase 2 clinical trials successfully. That means both programs are de-risked as far as safety and clinical activity is concerned. What we are trying to do is to formulate them differently such that they may have local action or superior action. And that is what New Biosciences is. Now, Tim and Lou, they don't need introduction. And but I must tell you this, and I mean it, that such a large credit for my past success goes to the center. Tim has been a mentor, very supportive and encouraging. And I still remember the early days of Synergy Pharmaceutical when we were in Death Valley. Tim was very supportive. As you see, this institute is transformed into a new heights. And I can say that Lou has been instrumental in moving this. So what I believe is with Tim and Lou helping me, New Biosciences is in good hands. As far as I am concerned, I think I've done well. I have bench to market experience. I was member of Celebrex. Many people know it's a multi-billion-dollar drug. I was the first one to explore Celebrex for cancer treatment. Eventually, Celebrex was approved for colon cancer in 1998. And I was heading the cancer group in Monsanto, in Central West. I am the inventor of Trulance. I also invented dolkenatide, which has completed phase 2 trial successfully. One good thing about this is that although I've been on board of several companies, but with Tim and Lou, I think we are in good hands. So while you consider this, look beyond on the potentials of New Biosciences. And what I'll summarize as, we have 2 programs, completely de-risked, validated with not only animal experiments, but also with phase 2 clinical trials. With that, I'll take some questions. Sorry, I had to go through pretty fast.
It's okay, beautiful presentation. I think we have time for 2 questions. Please.
Yeah, I may be showing my profound ignorance, but the first drug to treat inflammatory bowel disease, how do you get it into the gut without going through it goes through the digestive system, not being, stored in there? Or
I must tell you that you're not ignorant. Because this was the main question people would ask me in the beginning, it's a peptide.
Yeah.
Are you crazy to give it orally, it will get digested and all that? My answer was very simple. The uroguanil, the parental compound, is produced in the hostile environment of GI. So why can't I give it? So answer is, yes, it is stable to those proteases.
All right, time for one more question? Mike?
Yeah, excuse me if I didn't get a feeling as to what the market is, what your competitive market is, what you're trying to go up against, and what your potential return on investment on that focused program. And you have two programs going on, but I wasn't clear which one you were going with, and as Jack Palance said, pick one, okay, and that's up to you. You gotta pick one, and you gotta deliver that message to people so that they understand that an ROI is on the horizon within a year, 2, 3 years, with specific benchmarks that you have to achieve, whether it's preclinical, whatever it is. So I'm just saying that you spend a lot of time on the science, not enough on understanding what your market is, what your product is, that evokes a differentiator that you're gonna be able to go to market, and who's gonna be interested in licensing or partnering with you, or what's your exit.
So my good questions, unfortunately, I didn't have enough time to go through everything. But I must tell you at the outset that both those programs are my babies. So I feel both are very important. Would I have enough time or resources to bring them quickly to clinical studies? Answer is absolutely yes. And I have done that before in previous 2 companies. As far as market and other things, there were slides. I have to go too fast. But the market for ulcerative colitis and ulcerative colitis induced colon cancer are big enough. That is,
But what is your competitive product that establishes the market right now? And what can you differentiate to accomplish that? Okay, I'm sure you have a beautiful baby, okay, but I'm not so sure I want to invest in your babies. Okay, that's from an investor standpoint, okay? And I'm
So,
trying to help you.
Mike, you're absolutely right. Obviously, my babies are beautiful. But I can tell you that based on my 30 years of experience, both compounds have significant differentiation. Let's say, for example, for pulmonary fibrosis, that is nothing. Right? I have something which works in animal studies, and based on what I've done in the past with the compound, it has high chance of success. As far as ulcerative colitis is concerned, you have acetol or salicylate derivatives don't even work. Biologicals do work, but biological is very expensive way of thinking. My goal all my life, to my synergy or Tiziana, is to be able to come up with something that patients can take home, and that they can, cheaper treatment. For money making, absolutely yes. There is a lot more.
Second question, just a comment about whether is it better to have one drug in a company or maybe 2 drugs, more than one. I think that's pretty much subjective. It depends on the nature of the individual capitalist investor. I've met investors that only want one drug per company, and all the resources have to be focused on that. And other investors who absolutely require a backup plan, provided the backup plan is also as attractive as a lead molecule. I think in your case, what you have in favor is the fact that the 2 lead molecules are at least thematically related. I think you can probably, claim that you have anti-inflammation fibrosis company and that they are somewhat related. If they were completely different, you probably wouldn't want to put them together. So there is truth in both sides of the argument. Yeah.
All right, so for the sake of time, let's do applause here for For those of you in the back, we have plenty of seats up here, so feel free to sit down. And with that, I'm gonna pass the mic back over to Lou for a quick audible.
We're going off script, so, couple house cleaning, yes, please, if you want to sit down, please come up front. Each entrepreneur that presents tonight is gonna have a table out in the, when we're doing networking. So again, any other questions, follow up with any of them, each one of you guys will go out and you'll have a chance to discuss one-on-one. The other thing is with the microphone, please, as you're presenting, try to keep it as close up here as you can. Shane, you did a great job. I thought you were sword fighting with the microphone, but, and with that in mind, I'd like to introduce our another special guest from the county. Again, we're very blessed to have such great elected officials that support us. Commissioner Harvey is always there if we have an issue or concern, and he's always a phone call away. When I mean it, he does pick up on the first or second ring. Ladies and gentlemen, Commissioner Harvey.
Thank you, appreciate it. I know no one came to see me, so I'll be very, very brief. It is interesting. I call Lou and he never comes out. So I think we know where the priorities are. But I do love, I really do love coming to this place. My office is just down the street, because of the promise it has, because of the brain power that you have in this building every single day and the work that's being done. Having a child with an autoimmune disease, having lost too many family members to diseases like cancer, any medical breakthrough is something I appreciate deeply. And I know obviously I'm not the only one who goes through those kinds of things. So it's also a great ego check for me, because I consider myself a pretty intelligent person. I mean, my field is always the social sciences, but coming here and just sitting through most of Dr. Li's presentation reminds me how stupid I am, which is a good thing to keep that ego in check. But the county, on behalf of the other commissioners, are always happy to support any work that's done here. And really just everybody who's here and whether you're here just consulting and being that that critic, and being that person who asks those questions that sometimes make you uncomfortable. That's how you get better. That's how you progress. And it's necessary. So thank you for doing that. And those of you who are pushing the boundaries of what can be done and how we, can manipulate this mysterious body that we all have, to make things better and prevent illness and disease and give ourselves some more sunny days on planet Earth. We really appreciate everything you've done. So thank you so much.
All right, thank you very much. So with that, I'm going to pass the mic over to Jennifer Griffin to intro our next organization.
All right, thank you. I have the pleasure of introducing Advent Therapeutics, and specifically David Lopez, their CEO, who's going to tell us more about a really interesting opportunity developing a soluble formulation of vitamin A for neonates who have significant clinical unmet needs. David has over 30 years of biotech executive experience doing a variety of things, very impressive, from the CFO to general counsel to operations and manufacturing. So very significant experience. Most recently, he was a founding member of the Discovery Labs team, where he was the general counsel as well as executive vice president there. Before that, he was the executive vice president, general counsel, and CFO of Indiocom, very busy guy. He also has been serving. He is a specialized consulting services offered to Insmed and Otsuka, as well as in private practice for many years locally. So I will pass over to you, David, and tell us more about Advent Therapeutics.
The answer is over here. I'm not going to move around that much. I have my 10 steps in for the day. And Jennifer, thank you for that gracious introduction. And of course, I want to thank all of you and the Pennsylvania Biotechnology Center for this opportunity to talk to you about Advent's mission and the game-changing neonatal and pediatric therapies that we are developing. We move forward a little bit here. Advent has a singular focus on a novel, late-stage vitamin A-based metabolic and reparative therapy that we are developing as both injectable and breakthrough aerosolized, non-invasive aerosolized drug products, for which the concept has already been clinically validated. We're targeting bronchopulmonary dysplasia. Bronchopulmonary is also known as BPD. It is an orphan disease. It is a serious unmet medical need. It is often chronic, incredibly costly. And even in the face of really some significant advances being made in neonatal medicine, BPD rates are high. They're actually increasing. There is no approved therapies, and the rate of preterm births in the US has just hit a 15-year high. Yet our products are approvable in relatively short periods of time. Also, I'd like to add that we have previously been awarded $6 million in non-dilutive NIH funding. And we have been granted both US and EU orphan drug designations. And just this morning was able to announce that the US FDA has awarded us rare pediatric disease designation, thank you, which makes us eligible for the potentially very valuable pediatric voucher. All of this providing potentially streamlined regulatory pathways to approval. A bit more about BPD. As I mentioned, it is the most common serious morbidity of prematurity. It is strongly associated with vitamin A deficiency. However, providing safe and effective vitamin A supplementation to these really fragile preterm infants is quite challenging, which is why we are so focused on developing our novel retinol palmitate drug therapy. As you see in the middle of the slide, the causes of BPD. BPD actually has quite a large at-risk patient population, with 100,000 plus infants born each year in each of the US and EU and many more throughout the rest of the world. It's also incredibly expensive. The first year hospitalization costs in the US average $442,000 a year, with an average hospital stay of 103 days. Often quite longer. And I just think about that, what it means for the patient and the parent and the family. And the national healthcare costs for BPD in the US are approaching $2.5 billion. And BPD often leads to lifelong disorders such as COPD, asthma, and pulmonary hypertension and the like. We have validated already in the clinic the concept of BPD, vitamin A for BPD, and vitamin A drug supplementation, which is what our products do optimally, has been already clinically shown to reduce the incidence of BPD with a number needed to treat of 12 to 14 patients. What that means very simply is that for roughly every 13 BPD at-risk patients that you treat with vitamin A, one more infant could be expected to survive without BPD. And that is a very compelling number needed to treat for therapies that are routinely administered in the NICU. The evidence, strong evidence supporting vitamin A for BPD includes a large NIH-supported clinical trial that was published in the New England Journal of Medicine, a comprehensive Cochrane review that details 11 separate neonatal trials where vitamin A was used to prevent BPD, and even more recently, a Journal of Perinatology article supporting the use of injectable vitamin A and providing compelling healthcare economics for its use. And no less, as we leave the slide, the National Heart, Lung, Blood Institute has stated in print that vitamin A convincingly and safely prevents BPD. We are developing a novel metabolic and reparative technology. It's based on the all-trans form of retinol palmitate. We have optimized the formulation for both injection and non-invasive delivery directly to the target organ of interest, the lung. Vitamin A is a potent morphogen. It has a multimodal mechanism of action that not only addresses vitamin A deficiency, but also, of course, is helpful in preventing neonatal BPD and supports normal lung development. We have a straightforward pipeline. Our injectable product is approvable within 18 months, and our novel aerosolized product within 3 years. The injectable vitamin A is administered soon after birth at a dose of 5,000 IUs 3 times a week for 4 successive weeks. A course of therapy costs $7,200 to prevent a potentially devastating case of BPD, and that cost easily fits within well-established drug reimbursement guidelines for premature infants. These products are attractive commercial opportunities. The injectable product represents, in the US alone, a $100 million market opportunity, and the aerosol product represents at least a 3 to 4 times greater market opportunity of $400 million plus in annual worldwide revenues. You may question, why does the aerosol product represent such a game changer? It's very simple. It fundamentally changes the neonatal treatment paradigm with non-invasive delivery, again, directly to the lung. This allows potentially all at-risk preterm infants to be treated. And we collaborate on our NIH-supported programs with an internationally renowned key opinion leader and neonatologist, Dr. Verinder Rehan, and the Harvard UCLA Medical Center, the Lundquist Institute. We have international patents filed on both the novel formulation and methods of use, which encompass larger downstream applications, such as acute lung injury, ALI, hyperoxic lung injury, and even chemical biological assaults. I want to share some data that's impactful from our NIH program. It paints a strong picture of what vitamin A is doing in the lung. On the upper left hand is a normal, healthy lung. As you move down, you see the severe disruption caused in normal lung alveolar architecture from a established model of hyperoxic lung injury, which mimics BPD. Moving over to the right, you see the observable benefit that injected vitamin A has over untreated BPD. This is essentially the effect that is already validated. But moving up, you really see the dramatic effect that Advent's novel, non-invasive aerosol dosing has on, not only restoring normal alveolar lung structure, but also normal oxygen CO2 exchange. We have a full product roadmap with several important and valuable milestones, including launches of our injectable product and worldwide launch of our aerosol product. And we plan to commercialize ourselves in the United States, and we benefit from extensive experience in the neonatal marketplace, long-standing relationships in the neonatal medical community, including neonatologists, NICU nurses, respiratory therapists, and the NICU represents an extremely efficient selling model where a small, focused technical team can well target the 290 NICUs that are responsible for seeing over 90% of the patients we would be focusing on. And as we leave the slide, there is extremely strong health economics that support our optimized drug products. Again, strong team, and we have very valued and important partners that have and continue to support our success. We are raising $2 million in Series A preferred financing. The funds will be used to complete manufacturing scale-up and bring our lead product to market, and importantly, to advance the aerosol program to a very valuable IND-ready stage for worldwide approvals. We are offering a 20% equity interest for the $2 million round, and we have prior indications of interest of up to $500,000. The company has a very clean capital structure. Presently, internal stakeholders own 100% of this company. After the financing, investors would own 20%, and limited subsequent financing is expected. And in concluding, we have built a late-stage biotech that is reinventing vitamin A for neonatal and pediatric unmet needs. We're targeting a neonatal orphan drug opportunities with a lead product approvable in 18 months, represents an attractive commercial opportunity. We benefit from an extremely efficient selling model focusing on the NICU. We have a follow-on novel aerosolized program representing 4X home run. And increasingly, pediatric orphan drugs present strong partnering and exit opportunities. And on the bottom of the slide is just a handful of relevant high-value transactions in this space. And together with some companies that would make logical partners or potentially even acquirers of our technology. I want to thank you and certainly happy to answer any questions.
All right, we fortunately have time for some questions. Fantastic, succinct job. Yeah. So open up the audience, please.
First, I need a short nap and a little ice cream, but I'll be fine.
After the tech stuff.
You need some vitamin A.
So what is your IP situation and how do you differentiate versus what's being used in the market right now?
Sure. Absolutely on the point question. So the IP situation is we have two core patents filed internationally. One on the formulation, which since they're patent pending, I get into the details a little bit, but really a novel approach to formulating what is normally a pretty persnickety oil-soluble API, vitamin A palmitate, into a water-soluble formulation where the micellar structure is small, it's consistent, it's durable, and most importantly, it can be aerosolized with particles in the respirable range that are perfect for, as you can imagine, neonates are not that easy to basically deliver an aerosol to. It's not a lot of respiratory drive there. And we also cover some novel methods of use, as I mentioned. So not only with respect to the more traditional historic uses of the drug, but also with respect to things like, I think I mentioned ALI, hyperoxic lung injury, and a burgeoning area of chemical, biological assaults from, industrial accidents like chlorine gases and such. You had another question too, the differentiation.
Are they using it now in the marketplace in its natural form and how do you expect to differentiate from it?
So there is only one form of intramuscular administratable vitamin A that we know of. It's been approved for over 70 years. It goes by Aquasol A. So when I talked about the clinical validation, and I would never disparage anybody's product, it's an old product. I mean, times change, you optimize products in this industry, you do the right things. It's that product that produced those clinically compelling results. Now, what we have done is not only reformulate for a better acting product, we have removed, from the NICU standpoint, were some really problematic preservatives, chlorobutanol, which is an alcohol, obviously implicates neurodevelopmental issues for neonates. And we use a solubilization process that's very unique to our fragile neonatal intended patient population.
So is the PRV attached to the IM version or the inhaled version, or simply to the molecule?
You're talking about the pediatric voucher?
Yeah, the PRV, correct, the
Yeah.
voucher.
So that's to the entire spectrum, basically.
Okay, so
And our orphan designation, both US and EU, really covers the broad spectrum population wise.
Yeah, and actually a remarkable event from my experience, the agency came back and specifically told us, hey, we're granting you an orphan designation that's broader than what you requested.
That's great. So you'll get 7 years of market exclusivity in the US and 10 in Europe.
10 years in Europe, that's exactly right.
That's great. Any other questions from the audience?
Is the voucher limited to pediatric applications or is it for?
So can I comment, because I got a little bit of an education on the voucher recently. And it's controversial because it's supposed to stimulate the development of drugs for these, safe harbors that nobody is working in, these rare pediatric diseases. So you can either use the voucher yourself for a follow-on program, or maybe as equally popular you can sell the voucher to another player and they're quite valuable. I mean, there's more now than there ever was. But I think the current fair market is anywhere from 75 million to 100 million plus.
Yeah, I had a question. Is there a defined patient population that has a vitamin A deficiency? And do they do worse than the others?
Well, you know, the patient population, the preterm infant patient population that has vitamin A deficiency well exceeds what is considered to be the BPD at-risk patient population. So time not permitting, we didn't even get into that. So an intramuscular version of optimized vitamin A drug therapy, much less aerosol, would potentially almost certainly be used much more widely than just BPD at-risk patients. It just so happens that vitamin A deficiency is so strongly associated with BPD. It's also associated with a lot of other developmental issues, and as we could all imagine, disorders like retinopathy of prematurity, et cetera. So there's applications for this we haven't even gotten into.
Any other questions from the audience? Mike?
Yeah, vitamin A, if I go back to my pathology training, which was back when the dinosaurs were roaming, is not innocuous. It has toxicity in high doses. And one of the issues I say, you're gonna get approved so that the pediatric population has to be treated, it has to be tested for vitamin A deficiency, because if a pediatric or neonate does not have a vitamin A deficiency, is there a possibility of getting into toxicity? And what's the safety profile in terms of dosing? Is it a very finite, a small effective dose? And what's the toxic dose? Do you have all that safety worked out for this in non-deficient patients is where you'd be concerned about this,
Sure.
especially in pediatric patients?
I mean, the short answer, and I'll expand, is yes, we have. We've done a lot of work on that. And a lot of it has, quite frankly, been done by the reference listed drug, Aquasol A. Aquasol A has been approved and used for over 70 years. The existing FDA-approved package insert provides specifically for not only pediatric patients, but specifically for neonatal patients, recommended dosing guidelines. The guidelines I talked about for BPD that are well recognized by neonatology fit within those already existing package insert approved guidelines by a safety factor of 100%.
David, just one comment, by the way, very good presentation. And vitamin A is very lipophilic. So, delivering it to deeper parts of lung could be an issue. And you might want to look at.
Could you repeat the question? Because I don't think everybody in the back heard it.
Oh, okay.
Oh, no.
So vitamin A is lipophilic, and therefore delivering the vitamin A to deeper parts of lung could be challenging. And what efforts his company is making to make sure that vitamin A is delivered in the deeper part of lungs.
Robert, do you want to?
So let me respond to that. So in terms of the dosing that you're going to be using, because it's directly delivered to the target organ, we can essentially emulate the equivalent of the 5,000 units given 3 times a week. Our existing preclinical data already show, you saw the slide that Dave presented, no toxicity at that appropriate dose. We've done some early tox work also. Haven't seen any toxicity as yet. Again, at the doses that are being recommended per the guidelines and per the Aquasol FDA label.
All right, any questions from the panel?
I am very intrigued by your go-to-market strategy. So as a therapeutics investor, we almost never see it. But it's interesting that you're taking advantage of a very concentrated NICU model in this country. And so intellectually it makes sense. But I wonder why versus, partnering with any one of these large pharma on the bottom here that might be able to help you through the clinical process and obviate the need for go-to-market at all.
Well, I mean, part of it is maybe building to the DNA of the company a little bit. A large contingent of us comes from Discovery Labs for a small niche respiratory, disorder that's called RDS, which is 3 degrees of separation from BPD. We developed the most novel surfactant and realized at the time that, especially at that time, these are not attractive commercial opportunities necessarily for big pharma until you get them there. So we designed, and Jennifer's point is good. You're not really going into the NICU and commercializing. I don't think a neonatologist has seen a salesperson in probably 20 years. You see medical science liaisons, you see clinical nurse educators. So no matter what direction we go, we think building that internal tangible talent will hold us in good stead, whether we partner, whether we go with a commercial sales organization or whatever. That answer the question?
All right, yeah, just to echo what he said, we're currently in rare disease in the NICU, and you can do it with a low OpEx and investment and cover most of the NICUs globally. So cool, great job, and with that. Standing up for this one.
Yeah, I have to take my 10 steps. It is my great pleasure to introduce today the next company presenter, Nick Spring. Nick is a multi-decorated veteran in the life sciences. He's a true life sciences renaissance man of sorts. He has achieved a remarkable track record of success in big and small organizations alike, across pharma, biotech, medical devices, and clinical trial services, holding senior executive positions at Merck, Mariel, and Relief Band Technologies, among others. Apologies if I butchered some of these names, Nick. Along the way, he became highly experienced in the development and global commercialization of drugs, biologics, medical devices, nutraceuticals, and dietary supplements. He's a serial entrepreneur who has taken companies from their startup phase through financing stages to exit. For example, Nick was the founder, chairman, and CEO of Topaz Pharmaceuticals, a local company, which he sold to Sanofi for up to $200 million. Nick is also a senior partner with the Bracken Group, has served for 10 years as an adjunct professor at the University of the Sciences in Philadelphia, specializing in pharmaceutical, biotech, and medical device entrepreneurship, marketing, and commercialization. And another remarkable distinction with Nick is that he has been a friend of the center, of the Pennsylvania Biotechnology Center, since 2005. Today, Nick will tell us about Cerna Diagnostics, his new venture in the diagnostic space now. Cerna has developed a non-invasive platform to detect mutated RNAs circulating in the blood, hence the name, which promises to be a marked improvement over all other current non-invasive tests. The company's mission is to save lives by detecting disease early, when it is manageable or curable, and to guide therapy decisions. So let us welcome Nick on stage. Nick, your turn to take the 10 steps.
Good afternoon, everyone. You all hear me? Yeah. Well, welcome to the show that never ends here. So a couple of things. One, just to let you know, I flew in from Crete last night, and I'm like 7 hours difference to you. So I'm at about 11:30 p.m. So I'm using that as my excuse around up front. But as I said, welcome, my friends, to the show that never ends. I'm a passionate entrepreneur in the life sciences. I'm driven by the fact that we can make the world better for people, no matter what their state is. Now, I'm also an experienced entrepreneur and investor. Let me just make sure this is going to work. And you know, when anybody's ever looking at an investment, I think they're looking at three major fundamentals. First of all, is a great idea. The second thing you're invariably looking at is there a large market potential, an unmet need that could make patients' lives better. And the third thing is, do you have a great team who can really execute on those first two and make anybody's investment really ring true and get the project done in a reasonable time, in a reasonable way? And I believe with Cerner we have that. Now, what we're going to look at in the next 10 minutes, so it's going to be relatively rushed, is we're going to look at a vision, problem, the solution as we see it, the opportunity, our technology. By the way, there's some horrible puns that are going to be included in this presentation. The timing, the team, and our next steps, okay? So I've been working very closely with Ajaz, who's sitting there at the back, and Tim, who's at the front. And also, actually, I have to mention Colin Miller, because in the development of this, it mentioned I do work heavily with the Bracken Group, and we've been helping this company and a number of other ones with the spin-outs. So first of all, look at our vision. Our vision is actually relatively simple. We want to use circulating mRNA and variant mRNA as a new analyte platform for non-invasive disease detection and management. And as you're probably aware, there's 4 major blood analytes: protein, lipids, DNA, microRNA, and mRNA. But we can use the mRNA platform specifically for liver. And let's not forget that the Bloomberg Institute and PABC was originally, and still is, highly involved with things like hepatitis. And having Tim included in this team is a real ace to play, because this is a hepatitis or a hepatic play. So that is fantastic. So you've got liver cancer, HCC and CC. I'm not going to try and get my tongue around that, given the jet lag right now, the real long words. Other liver diseases, cirrhosis, and non-alcoholic steatohepatitis, NASH. Other organ sites. This technology has the potential not only for HCC, but also other types of cancers and detectable elements. So this is just the way that we get into the marketplace. So using, across the chasm, land and expand, this could be the first movement that we make as a company before moving into other markets. For identification of neoantigens by better biomarker databases, potentially for AI. And I believe, by the way, in fact, I know AI is going to be super massive in this market. I've been working with a number of different companies recently, especially in the real world data section of the market. Now, how big is this problem? It's really, really big in the USA. There's over 30,000 HCC liver cancer deaths in the USA in 2022, just last year. That's a crisis. We have 633,000 people with liver cirrhosis in the US, 3 million people with chronic viral hepatitis. We have 5 to 10 million people with NASH, and 30 to 40 million with metabolic fatty liver. And there you have the distribution across the USA. Okay, it looks even worse than the current heat map of across the USA. So it's a really, really big problem. And the big problem with hepatocellular carcinoma, ACC, is that the current detection methods are really, really lacking. Okay, and I'm not going to go into great detail. We've got 10 minutes, but our major advantage is that we will be able to detect disease very early on and get people treated early on, because the tragedy with HCC is it's invariably diagnosed at end stage. So people die. If you could get it much earlier as a standard test in a standard diagnostic with blood, you could really catch a lot of people really early. So that's really our general thesis here. So human circulation contains a diverse number of mRNA transcriptomes. And here's where the puns start rolling out badly. And I'm going to blame my colleague Tim over there for coming up with every one. So first of all, we got the Miracle assay, TM. And it's a novel non-invasive test for liver disease. So you don't actually have to go into the liver or do any bio tests or anything like that. You can just take straight blood. And it's an in-solution detection of mutated messenger RNA derived from diseased tissue. And it's providing risk stratification, early disease detection, and guiding therapy. And in fact, we believe that potentially there's also a development program. We could go into theranostics as well, but that's probably a story for another day. And we can do this from a platform technology that we can use for other disease detection. Now, if we look at the miracle tests, then we come into the second lot of terrible puns. We've got miracle splice girls and splice guys. And its text applies variants, but tubes in general and HCC in particular produce mutant mRNAs detected by the miracle assay system. So this will be detecting a basket of mRNAs, very early detection. You could then go into more specific if somebody came out positive. So that's basically our thesis, as I said. So detecting and characterizing these mutations in circulating transcripts offers a non-invasive approach for early detection of patients. And that's what it's all about. That's some mRNA on there on the left. And the human circulation has a diverse number of mRNAs. And these can be single nucleotide, post-transcriptional errors. And these variants reflect mutations in the tumor and may be undetectable except by RNA detection. So that makes us really, really unique. So you've got a big problem, a very unique detection method, gets people early, and here you have the potential for a huge success and huge demand for your product. Getting into a little bit of the science, and in any sciencey questions, you can certainly fire at Tim or Ajaz later on. But basically here we have, I think, quite a self-explanatory type of chart where we have various detection of the mRNAs, and obviously when you get to the HCC samples, you can see there is a very highly detectable area that makes it very clear about how we're detecting these things. And along the bottom axis, by the way, probably can't read from the back, are the variants that you could have in terms of biomarkers. So you get a high concentration where you have a disease. Going into it even further, you can go down to each variant biomarker and also the conformations of each. Again, I'm not going to go through that today in a 10-minute presentation. We can assure you in a further meeting that we could have, as potentially benefits to investors, we can certainly go into this significantly more. So what are the distinct advantages that we have over current systems that are out there? Well, firstly, we can detect mutations associated with LC, active cirrhosis, and HCC, and guide medical management early. We can detect mutant proteins that can't be detected by antibodies. We can detect proteins that aren't secreted, and we can detect splice variants, which, compared to detection of DNA, mRNA exists in many copies per cell, compared to a single copy of DNA, hence miracle, as a promise of greater sensitivity. And DNA in the blood is from every organ. Miracle can restrict analysis to liver-derived mRNA, hence promise improved specificity. So you've got something that has greater sensitivity and greater specificity, which is great for any sort of diagnostic. Now, of course, I'm from Britain, so I'm going to show you a castle with a moat and a little drawbridge. Well, actually, the ones. But we have already put provisional patents in. We have obtained exclusive licenses, and we intend to build a strategy and build a castle and a moat. I can already see Mike's getting so excited. He's talking to Tim and wants to write a check.
I don't want to shut up. I don't want to.
In terms of timing, we're already working forward with the ACC RICS panel this year, an outcome with our theranostics ideas as well. And we can move forward into 2024, and we've also got the NASH as follow-on. So that's really our overall timing element at this point in time. In terms of strategic plan, it was interesting actually the comment that was made regarding, go it alone. And when I've been advising companies generally, having run my own one, it's very difficult to establish not only a company in terms of getting a new product or idea out there, but also selling marketing is tough. So we've already made the strategic decision, and we were already discussing at the very highest level with some majors. And again, for those who are interested and want to go to an NDA, we can certainly talk to you about it. We have partnerships with major diagnostic developers already. We're in good discussions, and also major commercial labs who are out there every day testing large numbers of blood samples. So that's our strategy in terms of moving forward commercially. So we're going to license basically our technology to these other companies. I want to mention also, actually, just in terms of the money, we're looking that we feel that we can get the diagnostic developed for around about $4 to $5 million and probably do it in an 18 months, 24-month timeframe. Again, I can go into more detail with those who are interested. Now, the final thing is you really need a knockout team who can execute on the plan. And I know this because, I've run companies personally myself, and there is an old adage that if you've got an A team and a B idea, you'll get there. But if you've got an A idea with a B team, you won't. And we have undoubtedly an A team. Our president, Nick Pebble, is a PharmD and a JD, and he also happens to be a patent lawyer. That's a very useful person to have on any team. Chief Scientific Officer is in England. He'd have a knighthood, Sir Timothy M. Block. And we're obviously in this building here named after him. I think could be anything better, a world expert in anything to do with anything hepatic. We're very lucky in having a chief scientist in Ejaz, Syed, who's a super guy. I've got to know him very well over the course of this particular project. And he's got great experience, not only in this, but also previously in oncology as well. And myself, well, I've got my own track record. I can talk about that another time. We also have a really cool board of advisors and directors of the company. We've got Wayne Yetter, who's the ex-president CEO of Novartis Pharmaceuticals. He knows a bit about this business. We got Lou Kasser, who we all know. Sorry, he's also in another rival company here, but Lou loves us the best. We have Tim Block, obviously, and Nick I've already mentioned. And on our scientific and medical advisors, we've got Harvey Alter, who's not only got Alaska, but is a Nobel laureate. We got Robert Gish, medical director of the Hepatitis B Foundation, and Richard Pastel, who's also been highly involved with all things hepatic for a number of years. So we have an excellent organization. We, as I said at the beginning, a great company invariably has a great idea. Here we have a very simple, very specific diagnostic that has a high demand and potential. We have a very large market out there that needs to be served. And we've got an excellent team who can execute against this plan, is enthusiastic every day to bounce out of bed and get it done. So there you have it, ladies and gentlemen. Thank you very much indeed. We'd love to field any questions that you've got. Thank you.
All right, we got time for a few questions. Great work. Anybody?
So, Nick, how stable are these RNAs that you intend to detect?
That's an outstanding question. I also have to point out that, England's cricket team is doing very well at the moment.
We beat you all the time.
Lately, lost by 10 wickets. Anyway, I'll file that over to either Ejaz or Tim. Did you hear the question?
Yeah, they're stable. Actually, Aegis has examined that, and the markets moving are very stable.
Okay. Any other question? What about the panel?
They're stable in serum, in plasma. That's the RNA. Because
These are actually in extracellular vesicles.
they're extracellular vesicles that you isolate.
Yeah, question here. So of all these mutations that you're pursuing, are you making a product that will target all of them, or just one or two? How does that work? How do you envision that this will develop over time?
That's an excellent question, Lorenzo. I'm sure that Tim and HS can also follow up. But we basically have a basket of antigens so that we'll be able to detect, or biomarkers rather, disease very early across the board. We could then go into much more specific disease definition through diagnostics post that. But right now, just having something that could be used routinely as an HCC screen would take us a long way forward. And that's really when we've been talking to potential diagnostic partners, companies, and also large organizations along the lines of LabCorp and Quest, then they are very interested in this because they want to really add extra diagnostics to their spectrum of diagnostics they already have, which is why, again, on the commercialization end, you're feeding a machine that basically wants to expand their appeal to both physicians and patients. I don't know when Tim or Chaz, you want to add anything to that? Okay. Yes.
Could you contrast for me the difference between exiting a therapeutic development company, which is the first two, and a diagnostic? I'm not sure I understand the exit monetization at the end when you're successful.
Right, I wouldn't say that it's an exit, it's a more of an ongoing business model. We see ourselves as being the R&D engine to continue providing various licensable products to the commercial entities. So you really have two major categories. We will start with diagnostics because it's the lowest risk and quickest route to market. Theranostics, though, ultimately also offer various opportunities in terms of us being able to identify and label certain biomarkers as we move forward. So really we have a dual path forward in terms of commercialization, although we have a common platform to address both of those particular models. That makes sense?
It does in a way. I'm still trying to understand what the ROI would be for an investor.
The ROI, I think that's a very good question. I think it would be relatively large because the demand for this particular area is very large indeed. You have a very large unmet need in terms of detecting, in this case, HCC. We know that there's a large established market. We know that a lot of people are dying from this disease today, and we've got to avoid that. So I would think the multiple would easily be in the 10, 20 range at least, because you're going to get a long tail in terms of the licensing opportunity with the various companies that you're going up.
I guess along that same line, have there been any outcomes research studies done to show early detection of liver cancer and what that can say?
Yeah, there has. I mean, I just kind of Tim or AJ. I mean, the earlier you detect it, the better you're going to do.
It was summarized and there, we have a couple publications, but more validations needed. But yes, for early detection or risk stratification of liver cancer, it's quite solid, and obviously we think has an advantage over the current standards. For drug selection, that's being or the diagnostics, that evidence is much more recent. That's right.
All right, we have time for one more question. So, well, I gotta pick right there. All right, over on the corner there.
Maybe I missed it, even though I have no excuse, I wasn't in traveling like you were, but is this going to be a point of care test? Is it an automated test? Is it a LISA? Like, what is the actual platform to run this diagnostic?
Well, I think that's a very good question indeed. I believe that what we'll be doing is that it will be a point of care or a point of diagnosis. I mean, you're going on to LabCorp request. I would imagine that working with our partners, we would want to fully automate it at some point because that's largely how they work. I don't know if Tim or HH, you want to chip in a little bit more?
I think the model is currently a lab-based test. So and as the figures showed, the plans on the companies we're talking to are looking at it that way. But it would lend itself to a CLIA. But the whole technology is I think Barry asked, asked the question, is an acquisition model. And we've had a number of companies from the center and also that I've been involved in the past that get acquired and the whole technology gets picked up. So we're setting up really a plan and a model where we're validating this test. We are actually the first to introduce an AJAS, saying this, the first laboratory to actually show to determine circulating messenger RNA and use it as a diagnostic. So we have that knowledge, know-how, and now of course some of the patents follow.
Great. Well, just as a reminder, our entrepreneurs will be out at the high top tables if you have any more questions for them. So first, let's just give a round of applause for our panelists.
Let's give a round of applause for Grant Castor, please.
Yeah.
More items. I know we have food and beverages out there, but I'd like to bring up, I've introduced earlier, Dr. Randy Hyde, President of Bloomberg, to talk about a very special fund that we're raising right now. Dr.
Yeah, and this is about our Let's Get Kids lab coat story. Sorry, it's hard to speak after all those great presentations to go after. There's the information up here. It's a way we pay forward. I think we do a lot of really good things here at the Biotech Center. And judging by the interest, the attendance today, but also remember, we need to educate, encourage, and bring forward tomorrow's folks. And so by paying forward, it's tax deductible. All of you probably have tax deductions to do. Please pay attention. We have very successful programs here, and I just encourage you to support them. Thank you.
Randy, was kids here, the kid from the summer program?
Not, we have a stuff on it, but thank you, Randy. And Tim, great retirement so far, I gotta tell you. The last thing I'd like to do is, we can't do anything like this. I think we have a world-class events team. I really want to thank the events team, Maura over there, Tyler, Ed's out there, Megan, Elena, Cassie, great job, thank you. All of you that have checks and you're ready to write them, again, the companies will be right out there. Thanks, everybody.